Hi everyone, I'm Xinyi. It's my pleasure to introduce our method STACI. It's a method that performs integration of spatial transcriptomics data with chromatin imaging using a graph-based autoencoder. And it's a method that's broadly applicable to many spatial transcriptomics experiments, including any in situ sequencing methods or methods like 10x BCM or SLICE. And we applied our method to an Alzheimer's disease model. We identified there's a region-specific disease progression. And we also identified biomarkers that are corresponding to this region-specific progression, both in terms of gene expression and in terms of chromatin morphology. And this work is in collaboration with GB Shivashankar at ETH Zurich and Xiaowang at Broad. So first, I'd like to introduce the data set we're working with. It's a measurement performed by called the star map method, and the data is collected here at the cells group. So in the experiment, there's a measurement of the gene expression in each individual cells, and then there's also a measurement of where the cell is located inside the tissue. Additionally, there's measurement of the chromatin of the cell, and later on we'll show that this is actually very informative of the state of the cell. And because it's an Alzheimer's disease model, we also performed paired imaging on the same cells of amyloid beta plaque and tau tangles. So in the experiment, there are 4 mice at 2 different time points, 8 months and 13 months. And at each time point, there are 2 mice, 1 control and 1 AD transgenic. So it's a particularly interesting model for studying spatial signals because AD has some non-spatially localized disease-specific cell type, and that's the disease-activated microglia. So when there's a progression of Alzheimer's disease, this disease-activated subtype of microglia would aggregate around where the amyloid beta plaque is located. And this earlier study was able to identify this spatial signal by only measuring 3 marker genes. Now we have the access to 3,000 genes in a star map experiment and even more genes in a 10x BCM experiment. We potentially have the ability to identify more complex spatial signals with interactions of multiple different cell types or cell states. But this also requires us to develop a computational method in order to identify such a complex signal. However, it's still currently standard to look at spatial data by only applying single cell analysis method that takes in gene expression alone. So it's standard to just take the gene expression from the spatial transcriptome experiment, do a clustering, and then look at differential expression of the clusters. The spatial information is only used later on to plot the cells back into space and then see which clusters are close to each other. But we hypothesize that perhaps if we incorporate the spatial information earlier on during the clustering step, that if during the clustering we take not only gene expression but also spatial location of cells, perhaps we can identify more complex interactions of cell states in space. So there's another line of work that takes in spatial signals and they try to identify spatially variable genes. So in these kind of methods, they take one gene at a time and perform a statistical test to see if that one gene is randomly distributed in space or if they follow a non-random pattern. But the limitation of those studies is that they only look at one gene at a time. We want to see that if we take all genes at the same time, including their spatial locations, can we then perform the analysis on the cell level of how cell states form different patterns in different regions of the tissue. Additionally, we also want to take into account chromatin imaging, because chromatin imaging tells us many information about the mechanical microenvironment of cells. In fact, our collaborator has a previous study that showed that if you change the size of the cell in a particular way and hence changing the chromatin morphology of the cell, the gene expression of that cell always change in a very predictable manner. So because chromatin imaging is also informative, we then take into account all 3 modalities in our method: the gene expression, the cell adjacency, and the chromatin images. And we put everything together into a graph-based autoencoder. And to make sure that this autoencoder learns all 3 modalities, in the output, we also decode all three modalities. And then we can take the joint latent space that has the information of all three modalities and do the usual clustering and to see which cells are close to each other, both in terms of gene expression and in terms of their neighborhood. And the plot here is the joint latent space. It's a UMAP of this integration of the all three modalities, and we can then take this clustering in the joint latent space and then see see how these different clusters are located in space. So the first thing we can see here is that each cluster is very consistent across different mouse. So if we see the purple region, which is the CA2, CA3 region in the mouse hippocampus, then the purple region is always the same across all the different bytes, despite of the batchy bytes that are coming from both gene expression and tissue morphology. And if we look at the reference slide as well, the clusters are not only consistent across different mice, they're also consistent with the reference slide. That there's one cluster for DG here in green, and then there's one cluster for CA1, and there are some clusters for the cortex region of the mouse brain, and that's all. And all these clusters are highlighting comparable regions across different mice. So there's also one thing interesting to look at this plot, which is that the cortex clusters here group the mouse cortex in a non-conventional way. Now, if we look at the reference slide, it's usually this horizontal layers, or there's some vertical divide as well. But then it's not immediately obvious why these orange why these orange cells would be grouped together, and why there's this divide here in the cortex. So it's actually corresponding to how different regions are whether the different regions are more advanced in the progression of Alzheimer's disease. And additionally, we also used a non-standard autoencoder in the sense that usually autoencoder taking input data and project the data into lower dimensional space in order to preserve the most essential information. But we use an over-parameterized autoencoder in which the dimension of the representation is actually larger than input dimension. So if the input dimension have 2,000 or 3,000 genes, the latent space actually has 6,000 dimensions. So it can learn the identity function, but we will show that it actually does not, and it actually performs a very useful function, which is to remove the batch effect. So first, I want to show that the different regions in the brain, the different regions that we identified in the brain actually correspond to the disease progression. So on the top right, when you see how the three clusters divide the space in the cortex, you can already see that cluster 1 correspond to regions where there's less plaque, and the plaque are the things that's in white. So, and in cluster 3 region, there are visually already more plaques. And we also have a quantitative measurement of what are the size of the plaques in different regions. And you can see that for both 8-month 80 transgenic mouse and the 13-month 80 transgenic mouse, it's always true that in the cluster 3 region that there are more plaques and there are larger plaques. So, so it's interesting that our method identifies these regions that are not otherwise identified before, and these regions nicely correspond to whether these regions nicely correspond to whether the region is more advanced in the progression of Alzheimer's disease measured by the accumulation of plaque. And as I mentioned, like in a standard autoencoder, you would have a bottleneck layer where the input dimension is smaller, where the input dimension is larger than a latent representation. However, we used the autoencoder with an over-parameterized latent representation with 6,000 dimensions comparing to the 2,000 dimensional input. So if instead we use a standard autoencoder with a bottleneck layer and keep all the architectures the same otherwise, we will actually not be able to identify the consistent clusters across different mice. So here in blue, you're seeing all the cells in the same cluster, but the cells in that same cluster are mostly in the DG region, in the 13-month control, but they are outside of the DG region in the 13 months AB if you're using a standard autoencoder. So it's especially important that we correct for batch effect in spatial transcriptomics data, because although we have many batch effect correction methods for single cell RNA-seq, they only deal with the difference due to gene expression. But we now not only have batch effects due to gene expression, we also have batch effects that are due to the difference in tissue morphology, either because individual differences or because of experimental handling. So our method is saying that regardless of what the source of batch effect is, you can always just take the latent space and make it larger, and all the batch effects will be removed. Additionally, I want to show that the neighborhood information is essential for identifying the spatial regions that correspond to the accumulation of plaques. If in the input of the autoencoder, if we remove the cell adjacency from the input and also the output, we will not be able to identify the spatial regions. You can see all the clusters are mixed in the cortex region, and they actually correspond to the different cell types. So if instead you are interested more in identifying the cell types from different clusters, then perhaps you can use our model with just the gene expression as the input. But to identify spatial regions, it's essential to input the cell adjacency. So now we also want to take in chromatin images. And the first thing we want to show about the usefulness of this chromatin image is that if you already have some spatial measurements on some samples, and we have the gene expression, cell adjacency, and the chromatin images. Now, if you want to add one new tissue sample, you don't have to measure the spatial transcriptomic on that. You can just take the chromatin images and then predict the gene expression from the chromatin image. So if you have this new sample that only measured for its chromatin image, you can use the encoder. You can use the image encoder for the chromatin images to embed them into the joint latent space, which is a representation for all three modalities, and then decode the gene expression from the joint latent space as the prediction of the gene expression of this new tissue sample without spatial transcriptomic measurement. And the predicted gene expression looks very much like the real gene expression that if we leave one sample out during the training of our model, that our model has only seen the gene expression and cell adjacency of the 3 out of the 4 mice. And now if we only use the image of the 4th sample as the input without using this gene expression, we can first see that all 4 samples are very well mixed in this UMAP, that the predicted gene expression look like the real gene expression. We can then do the usual clustering analysis on the predicted gene expression and identify the cell types from the predicted gene expression. So we can do the usual cell type identification and then look at the markers of each cluster to identify which cell type is corresponding to different clusters. And the cell type marker identification are recovering the genes that are known to be the markers of excited neurons, oligodendrocytes, and DG, which correspond to the 5 clusters of this predicted gene expression of cells. So that showed us that the gene expression is very informative. Sorry, the chromatin image is very informative. And we want to do more with the chromatin images to identify perhaps the biomarkers during this disease progression and during the region-specific advancement of Alzheimer's disease. So, so now we have the joint latent space of the different of all 3 modalities. And we're interested to know that what makes the cell, like what are the differences in cells that are closer to the amyloid beta plaque, and what are the different, and comparing to cells that are more far away from the amyloid beta plaque. So we take the joint latent space of the different cells, and then use that as the input to train a regression model to predict the size of the plaque. So here's the result of this regression model, and the goal of this is not to predict the size accurately, but more to look at which region of the input image that are predictive of a plaque nearby. And if we look at the image on the right, so the green thing is the amyloid beta plaque, and the cell chromatin are in red, and the blue is a measurement of through activation mapping of how predictive that pixel is to the prediction of plaque. So if we look here, this microglia is, for example, very predictive of this plaque nearby. But if we look at this inhibitory neuron, it's all very low intensity blue. It means that this inhibitory neuron is not yet affected by the plaque nearby, and it's perhaps not very different from the inhibitory neuron that's further away and not affected by plaque. So there's the cell type differences, but it's also interesting to notice that the blue regions are not only highlighting individual cells, it's also highlighting specific regions in the cell chromatin. So if we look closely here, for example, in this excitatory neuron, the blue is only highlighting this region with very dense chromatin inside the excitatory neuron. Or here, if we look at the inhibitory neuron, it's also highlighting the region with very dense chromatin. The hypothesis is then that perhaps this denseness, or what we call the heterochromatin of the cells, was actually predictive of the plaque nearby. So this leads us to the next analysis, which looked at the heterochromatin versus euchromatin ratio of the cells, i.e., how the denser region of the cell chromatin and the less dense region in the cell chromatin. So first, we compare just broadly for each cell type, how the AD transgenic mouse compared to the control mouse in terms of their chromatin intensity. And you can already see that the control distribution of chromatin pixel intensity is mostly a unimodal distribution. And when it goes to AD transgenic mouse, that there is a shift towards higher chromatin pixel intensity, and it starts to become a bimodal distribution. So some parts of the cell start to be more affected by the plaque nearby and by the disease progression, and start to have a denser chromatin, and then having a higher intensity here. We can summarize this heterochromatin versus euchromatin ratio by looking at how that compares across all different cell types. So you can see that the heterochromatin ratio inside the cells has this shift to a higher value, going from the 13 months control to 13 months AD, and this is also true in the 8-month samples. And this is interesting in contrary to the general trend that going from 8 months control to 13 months control, there's a decrease in heterochromatin ratio. So Alzheimer's disease reversed the trend of aging, that it's now making the cells have more heterochromatin. And because I mentioned before that we hypothesized this heterochromatin ratio is related to the predictiveness of plaque, we also directly associate the heterochromatin ratio in each cell to the predictiveness of that cell to a nearby plaque. And you can see that in all the 3 different cell types, regardless whether it's neuron or whether it's glial cells, there's always this significant positive correlation of the heterochromatin of the cell and the predictiveness of that cell to a plaque nearby. That with a higher heterochromatin ratio, it's more predictive of a plaque nearby. And we can take this we can group the cells based on their heterochromatin ratio and whether they're predictive or not predictive, and group them into 4 different groups. So we can then look at these 4 different groups and see how this ratio of the cells in different groups change across space and across time. So this example is shown for microglia. We can see first that in the region that's less advanced in terms of AD progression, i.e. cluster 1, there's a less amount of shift of cells towards a higher heterochromatin. And but going from cluster 1 to cluster 2 and 3 in the top plots here, the AD transgenic mouse show a general shift towards the right, that the cells start to have more heterochromatin pixels in general, and that's true for both 8 months and for 13 months. And microglia is known to have an early onset of Alzheimer's disease. So even in cluster 1 region, which we said is less advanced in AD progression, that it has less accumulation of the amyloid beta plaque, it's already having cells that are high in heterochromatin and also high in the predictiveness of the plaque. And if we further look at what are subtypes of that cell that are predictive of plaque and are having high heterochromatin, and the green thing is the one subtype of the microglia that's known as the disease-activated subtype. So this is consistent with what we know about the Alzheimer's disease, that the microglia has an early onset, and this early onset is manifested in this disease-activated subtype. And if we move further to the regions that's more advanced in disease, we can see even the subtypes of microglia that are also observed in healthy samples, they start to have high heterochromatin ratio, so that they are more affected by plaque, and they are also more predictive of plaque in our regression model. We can then relate this biomarker that's given by the chromatin morphology to identify biomarkers of gene expression. So if we compare cells that have high heterochromatin and are very predictive of nearby plaque to the cells that have low heterochromatin and are not very predictive, we can identify genes that are associated with this change in predictiveness and in heterochromatin. And interestingly, the genes we identified here, a lot of them are not otherwise identified in a standard differential expression analysis between control and AD transgenic mouse if we do not take into account heterochromatin and the predictiveness of plaque. And even when we do a standard differential expression analysis between cells that are close to plaque and cells that are far away from plaque, there are many genes we will not be able to identify by if we do not separate them by heterochromatin ratio. And these genes are also also annotated in biological processes that are known to be important for the Alzheimer's disease, including secretion and transport. So to summarize, we show the benefit of a joint clustering of all three modalities. That is, in analyzing spatial data, it's important to take into account not only gene expression, but also the neighborhood of cells, and that we showed the chromatin imaging is very informative of the cell state. We can use them to predict gene expression. We can also use them as an identification of biomarkers and lead to a stronger signal when we analyze differential gene expression analysis. We also showed that there's an automatic batch correction for gene expression and the morphology across lines, and this is done through over-parameterization. And this corrects not only for gene expression and also for morphology. And then our method is also compatible with other platforms, including 10x Specimens like Seqfish and Merfish. If you're interested in trying our method out, it's on the GitHub. We have one notebook for in situ sequencing data using StarMap. We also have one demonstration using 10x Specimen. And it's a demo on the demo data of 10x Specimen with the brain samples. We show that the joint clustering can be used to identify brain regions that are at different stages of disease progression. There are some regions that are less advanced in disease progression. There are some that's more advanced in disease progression. And that's only using the gene expression and then the spatial location of cells without any knowledge of the accumulation of plaque while doing this clustering analysis. Additionally, we showed that chromatin imaging is very important biomarkers and we can use them to predict gene expression. And that's all I have for today.
Thank you so much for a very exciting talk, combining so many data types. Very, very cool. I want to welcome anyone in the audience here to raise questions, and the same goes for anyone, of course, listening online. I have a general question just about the accumulation of heterochromatin. Naively, I would think of that as a cell using less of its genome overall. And in addition to that, of course, you've got the enrichment analysis. And I wonder what your sense is of what that means. Are those cells just doing less because they're using less of their genome? Or
I'm not sure about the part that's using less of its genome, but then like people think that a general trend of aging when it's like less heterochromatized, it actually means it actually means that the cells are regulating the chromatin less. And the hypothesis is that because it's less heterochromatized, the gene expression is less controlled, and that relates to many aging-related diseases. So I think maybe this is I don't know, maybe it's not now actually more highly regulated, but I'm not sure about whether the regulation versus using less of its schema.
Thank you so much for a wonderful talk. One of the things that often comes up as we're preparing single cell or single nuke data sets is sometimes the initial sample that was taken for the RNA sequencing, you don't know exactly which face of the tissue sample it came off of. And then so you try to cut your slice to do your RNA scope or whatever off of the same face, but it might actually be a totally different aspect of the tissue, which could have a slightly different cellular composition. How much does that lack of continuity from where the single nuke data was generated from versus the spatial transcriptomics? Is it sensitive to making sure that they're immediately adjacent, or is millimeters or centimeters apart okay?
We're not actually using single cell data here. It's just the spatial data. So what are the...
So I guess if you were to integrate in the future, like the number of genes that you consider is somewhat limited by the number of probes you can do, right? So I guess I was imagining a future where you might use the higher dimensionality of single nuke data to maybe enhance some of your correlation. Sorry. And so I guess the first question would be, have you thought of doing that? Or is it just too difficult given the dimension of data from the spatial transcriptome?
I guess it's not difficult because we've also tried our method on the 10x museum, which has the full transcriptome. Like using single cell data would then be a different line of work where people are mostly interested in like deconvolving maybe 10x museum data. But it's an interesting idea to incorporate single cell RNA-seq with the spatial data because perhaps it has a higher dimension and it can give us a more fine-grained clustering because now we have more genes. It doesn't have to be that adjacent, so you can already see that in our data. You can already see that here. The slides are already morphologically very different, like the DJ region there versus here. They're probably slides at very different regions, so it doesn't have to be that adjacent. And that's one benefit of using an over-parameterized autoencoder because it corrects for this tissue morphological differences, and it also corrects for differences in gene expression. So it doesn't have to be that it's exact.
Thank you. I found that like over-parameterization and removal of redshift very interesting. Do you have any number of parameters you set? For example, if you're targeting, let's say 3,000 features, you're setting like twice or three times of that, or like is there a ratio, or like how do you select the number of parameters?
Yeah, that's a great question. So usually it's not very sensitive to the exact choice of the parameter. So if your input data is 3,000, you can keep trying to go higher. And so once it starts working, then it doesn't matter if you go even higher. So like if we start from 2,000 and we tried 4,000 first, we realize that it corrects some batch effects, but it's not enough. Then we go from 4,000 to 6,000 and realize that that's actually good enough. We also then tried further to go to 7,000 and 8,000, and they are very similar to each other. So once you're over-parameterized enough that you have to go to a dimension that's high enough, then you can keep going higher, we can stay there. The exact choice of the parameter is no longer making a difference, which is also good because we don't want too much hyperparameter tuning.
Great. Well, I want to thank you again for a very exciting talk and for hearing where your work goes in the future.